Um, we've been working really hard the last 8 months to convert our general chemistry and our organic chemistry into fully online versions. But also at the same time, we've been creating a back-end, what we call a portal infrastructure platform to manage these labs, look at lab books, and give you a lot more flexibility in how you do assignments and stuff. So we kind of went into a semi-code freeze just yesterday. So we have a really good stable platform. And if you could send in the chat, put your email addresses. I think next week we can have some people reach out with some trial codes so we can get you access to the platform. Recognize it's beta still, but it's a pretty good beta, and let you guys kind of play around with it and get some feedback from you. But this intro was really useful for me, so kind of know how to address things. So let me do this. It looks like we got a pretty even split of organic and general. Let me kind of do this in 3 phases. Let me kind of show you, for those of you who are using the older version, kind of show you the new platform a little bit. The labs really haven't changed. We've added a few new features, like for organic, we've added some multi-step reactions in there. For quantum mechanics, we've added some crystal violet kinetics experiments. And so there are a few new things, but the labs generally are the same. So let me kind of show you the platform, what the platform will do, how it works. If you're familiar with the labs themselves, they really haven't changed that much. And then let me, for those who are new, kind of give you a brief overview of some of a couple of lab benches that we have for general and organic. And then after that, we can maybe come back to the platform and show a couple of more detailed features. Feel free to answer questions or to shoot some questions in the chat for Heather. So essentially, we've had, these virtual labs have been around for almost 2 decades, really over 2 decades. We developed them started development in 1998. So by the way, I'm Brian Woodfield. I'm one of the co-founders of Beyond Labz. I developed the labs, and I'm a professor of chemistry at Brigham Young University. I've been there for 24 years. I'm a physical chemist. I teach general chemistry for majors and non-majors. And so I kind of invented the labs when I was a grad student at Berkeley, and we developed them at BYU. So they've been used a lot. They just kind of transformed over time as tech changes and bandwidth on internet changes. And so you're really the early people to see the full complete package of what we put together the last 8 months. So if you knew the old versions had a portal, but you had to download and install on Mac or Windows the labs, and they ran locally. And so what we've done is we've converted all of this into HTML5, so they're all browser-based. So we still have logins. We're still going to have codes. The codes are going to be a lot easier to use. The one thing that we're going to have is we're going to have a student role and an instructor role. So as an instructor, you'll be able to see and do things that the students won't be able to do. But if I had to say overall, one of the biggest improvements we've made besides converting the HTML5, it's lab book and presets and assignments of what we call presets. So
Brian, could you make your screen full screen to make it a little bit bigger for us?
Sure. How's that? Does that work? All right. So this is our new portal. We have a desktop side, so we haven't converted biology or physics or physical science. So they're going to live over here. I'm not going to focus on that because everybody here was pretty much chemistry and organic. But you will still have access when you purchase codes to those other products. So what we've done, instead of packaging them into chemistry, we just have lab benches now. So if I come over here and I click on calorimetry, so we're going to come load the lab, and so here's calorimetry. And so I got to minimize this a little bit. Let's get it out of the way. So you'll see this is the same calorimetry lab, but what we did is we took out the stockroom and we moved the stockroom over here on this tray. So it's all one page, don't have to go in and out. So I can come over here, pick a coffee cup, come over here, pick a metal, like gold, you know, and then it's still this open-endedness, still works the same way. Come over here, fill this up with some water. And so same lab interactions. So we'll add this, turn on the stirring, we'll turn on the thermometer. So we have, now we show all of our data in one of these tabs called live data. So I can come down here, I can come over here and tare and weigh. And so now I know what the mass is, zoom back out, open the oven, turn the temperature up to 200, and put this in here, close it, open it. So all of this lab stuff works the same, but what's different here is now the graph is all kept inside of here. I can go grab my metal and drop it in. I can save data here and save this data. I can save this graph, and all of that is going into this lab book. And so we can still accelerate time. So here's just a simple heat capacity of a metal experiment. Of course, I'd want to have it go further, but just in the interest of time, I'm not. Here's this data. So I can go to my lab book, and here's my lab book now. So notice I've been collecting data in my lab book. One thing we've noticed, so one thing that we've changed in the lab book is now this is in lab book is integrated, and we keep track of every time a student enters and closes the lab with timestamps. Every time they save an image, they save data. So we're putting these timestamps in, and here's the data that we saved. And you can copy this data so they can analyze it. They can come in here, put in notes. They can format those notes. They can convert this into a PDF file. But unlike the last version where the lab books were this external file that you had to move around, that's now integrated. You can also come here and show all the results from all of the different labs, all here in the same
Brian,
locations. Yes.
Are you acting as an instructor or a student as you're showing these to us?
So I'm an instructor, but the lab itself is going to work the same as you're an instructor or a student. What I'll show you in the back end is kind of what we see. I just kind of wanted to show you the basic idea of how the new labs are going to work. With tabs, we have this ability to do unknowns. So here I can clear the lab, come over here to this unknown. I can take a salt, create a salt unknown, and then I now have an unknown. And this is for practice, so I can just reveal it so they can go here and practice. This is only for pedagogical practicing for the student. But what we can also do is we also have presets. So the last lab had presets. These are the same ones. I can click here and there's my presets, and I can just click and go. But the nice thing is that I also can save my own presets. So here is a calorimetry known reaction that I put together. So here's one with sodium nitrate. It's known, and I put this together, and I can click down here and click Save Lab and save it. All right. So, you now have that really cool ability to save these presets. And the nice thing is that you can change their titles, but you can also copy a link. So I can copy this link right here. And let's say that I come out here and I actually sign out, okay, of it. I can come over here and just take that link and put it in. You can email it to your students. The students can email it to you. And I can go click return. It's going to launch it. It's going to say, hey, you've got to log in. So I'm going to go log in. And I'm going to launch that lab. And I can launch that lab and it will start with the configuration that I had before. So here's the lab configuration. So now you can take presets and pass them around as links. And you could also come over here and add that preset. So let's pretend it's somebody else's preset. You could add it, and now you've got a copy of it, and you can add it to your own personal list of presets. So I'm not really trying to show you, to those who are new to the product, what the labs can do, but this is what's new in terms of live data stockroom and unknowns, lab book, and presets of what you can do. So if you come back here to the portal, what you do is, this is where you can go and launch these individual products, lab benches that we have. So these are the 7. By mid-August, end of August, we will add density to the list. But Labs Plus, so this is what we call our presets. So you notice that in the tab, in the tray, in the lab, we had presets, but now this is where you can see all the presets for all the different labs. So if I want to do a cyclohexene hydroboration,
and these are the presets we made. There's all, yes. The other tab shows all the ones that you make and that you save. So we've got all of them there.
So here is the hydroboration one. So it's all set up, ready to go. Here's your flask contents, lab book. And so these lab presets are what we call system ones. So they're available to everybody. And we have like 93 of them. So you can come over here and sort, hey, I just want to look at the gases ones. You can look at gases. And then these become the ones you made. So you can come here to the portal and see these. So this again, Steve, so it's a good question. I'm an instructor. So an instructor and a student, this is going to look the same for them. But what's different is when I come to lab book. So when I come to lab book, what I've done is I've created a class, and I'll show you this in a minute, and you can see different students. So here's their names, and you can see when the last time they put some entries into it, and you can actually look at their lab books. So I can click on Josephine here and come over here and go up. Here's my mass spec and NMR and TLCs. So here's all stuff that we've collected for them. So you can actually go through and look at all of their labs, or you can go and just look at their synthesis entries. And as an instructor, I can come here and say, good job, you should major in chemistry, right? And so you can put these instructor notes. And the fact that I was in synthesis means that this student, Josephine, will only see it when they're in the synthesis lab, or you can come back here and show it in all of the labs.
I was just going to point out, hopefully it's useful to you. You can see how long they spend in the lab with all these timestamps. So you can see if they actually were working for a long amount of time, but you can also turn off the timestamps if those are distracting in any way.
Yep. So in here, you can turn off the timestamps and just look at what they did. So, so this is the nice thing. So you can go back, you can go through here and look at these students. The reason for this is, I don't know that I can't remember if I added one in this student or one of these. No, that one doesn't have a lot. But you know, you can create unknowns, like someone mentioned that they're interested in qual. You know, we have unknowns for titrations, inorganic, calorimetry, and for qualitative and organic. So students can report their unknowns to the lab book, and this is where you would go through and look at them. You can also look at your own lab book. So Steve, I'm an instructor, but I can still look at my personal lab book right here. So if you remember the old product, we had activities, but they were PDFs. So Heather and her crew have spent many months converting all of this content into, digital format. So here, so it's the same content that you're used to, except now when you click on it, it's in a digital format, and you can come over here. And so this is not recorded anywhere. What a student does is they would put in their answers here, and then what they would do is they would either print this out as a piece of paper, or they would print it and save it as a PDF. So they can save this document as a PDF version and then submit this in your LMS. We also have these exactly the same content, these activities, in a format that you can integrate into your LMS through LTI 1.1 or 1.3. So you can also integrate these, and here the answers would be graded automatically. About 80% of them would be graded, and you could record all of these results. So the way
that's
this
what Basil
works,
was asking about, right? So Basil, that's where, yeah, you can have all this content and just integrate it right in. So all the grades will go in your grade book, and it does it perfectly.
Yeah.
And the nice thing is that here, you remember in the old lab, it would have, start virtual chem lab, go here, click this preset. Now all you have to do is click on that link. I already clicked on it, so that's why it was purple, but I would just click on that link, and I'm going to start my titration lab, and it's going to be configured like before. I don't know why it's so slow. It's a little, my daughter just came home, so she could be stealing my bandwidth, but here you go. Here is this preset. So now you just come over, what you do is you come over here, you just follow your instructions, and you flip over here to this tab, and you go back and forth to go do it. So this is, so now we have this. So now here's a feature that we've added that is new. So all of this was kind of there, but we've kind of upgraded how we do it. Now we have the ability to do assignments. And this is, you don't have to do assignments. All you have to do is say, here's an activity, go do this activity. You can create your own presets and share those and say, here, use this preset and write your own activities, that's fine. But we want to make sure you understand the difference between an activity and an assignment. An assignment takes an assignment takes takes one of your presets, either our system presets or one of your presets, and puts a due date, a release date, and a due date on it. So here is one, for example, that I put together: inorganic chemistry, full cation, unknown. All right. I assigned it to a group. I'll show you how you can create groups. So I can have a group of students, and then what you do is you come over here and you pick a preset. And this particular one is I can convert a practice unknown where the student can see the answer and convert it into an assigned unknown. And I can have a release date and a due date, save it, and so on. So here's an example, right? If I click on this, then I'm going to start the inorganic lab, I hope.
And students will see those. They get this cute little red dot when they open the lab. Oh, you have an assignment. You have an assigned unknown. And so they know, oh, I need to go into that. And then it reports it all back to you.
And so notice here that this is what she was talking about. Remember, all the labs with the unknowns all have this unknown option. And so here is this assigned unknown, and they can't see what's in it. So what they would do is, they would add their acid, go add their chloride, and centrifuge this and decant, and they've separated. Now they've done a separation, and they would go through and do their work. And then when they're ready, unlike a practice unknown, where they could go here, this is an assigned unknown, and this would list all of the cations in their set. And looks like I know I have chromium because of the color, so I'm going to put chromium.
And this one happens to list all of them if you'd set up the preset to only be between like 2 cations,
Yeah.
that's all it would list. It customizes to however you set up your preset.
So I would submit this, show their actual results, and then when you go to the lab book, there is the lab book. Now, I'm an instructor, so I actually did this for me, but the student would see that. So they would see their response, they would see the actual result so that you could come grade it. So we also have, like, again, in these presets for this one would be like Heather said, something more simple would be like chlorides. If I click here on chlorides, this is a pre-programmed unknown for you that you don't have to do anything. All they have to do is come over here and click on chlorides, and here's their, come over here to live data, see what their test tube is, go add sodium chloride, see what it is. When they're done, they would come over here and, you know, I'm just guessing here, click submit, and there you go. I was totally wrong. That gets saved to their lab book. So when I come back here, to my lab book. If this was a student unknown, I would see these results, and you can go and give them a grade. So, so these assignments here, come back here to assignments. They're really easy to create. You create new, you put in some text, you put in a description, you know, right here, you select the group that you want, and then you come over here and you pick, oh, which of all my labs? Maybe I want a calorimetry one. So here's a calorimetry reaction unknown. You know, you come and you can make that an assigned unknown, and then you put in your release date and your due date, and you publish it, and you're done. And then they show up here. This is where you define groups. So all a group is you create a group. All a group is you give it a name, and it's going, and you can, we generated code, or you can type in your code. So like, I'm not teaching this spring, but if I was, this is what it would be. Some code you can, and then what you can do is copy that code and you share it with the students. And then what a student would see here is, if I come over here and log in as student 1, then you know, what they would see here is they would come, and here's the group, and you can join a group by clicking on this, and then they would see what assignments, their release dates, their due dates. You can come over here to get more information. This is just a preset. You would just click on it and go launch this. So anyway, that's kind of the overview of what the new platform can give you. Any questions or any comments you may have before we go on?
Yeah, I have one. Like, when you create the assignments, and if you are fully integrated, so we're using Canvas as an LMS, so those assignments will appear on the LMS, correct? And then you can grade them here or not. So how does the integration work?
So good question. So at this stage, these assignments do not show up. They're only going to show up in what we call this portal, this platform. It's these activities that will show up inside of your LMS.
Because the assignments are just like a website link. They're just a link to a starting point. They don't include the content that you would want to provide to your students of do this, do this, do this, do this. All of that is what an activity is that's going to then live in your LMS. You're going to assign them, hey, look, this is all the content I want you to read. Now click the link. And then the ones that I've written auto grade, and all the grades go right on in.
Okay, yeah, because with online, that's really the confusion that we had with students is, especially when you're teaching online, is like having a multitude of different like structure and platforms just is uber confusing for them. And you just have like always the one that like, oh, I had no idea. It's not on my LMS. I didn't know how to do it. It doesn't show on my calendar and blah, blah, blah. So that was really like.
Yeah, so if you use our content, they won't ever even need to go to that portal. Unless they really want to. They can just go straight through the LMS. They'll click the link there. It'll open the labs. I mean, they'll have to like log in to create an account. right, to get activated, but then they'll do all the work in the lab, report it all in your LMS. You'll do all the grading. You'll see the lab books though in our platform. The lab books, you have to go into our portal as the instructor to see. You won't see those in Blackboard, but the all of the answers to any worksheets would be there in your Blackboard.
Okay, and do we have to, for integration, do you have to enter in touch with our D team, or it's something that the instructor can do at their own level? Do you have
IT department, unfortunately, yeah, so you just need to send me an email and I could set it up with your IT department, and I just send you a file and you've got it. They can link it up on the back end and then we can walk you through it.
Okay, yeah, I'm part of the D team, so I can actually, okay.
Oh, if you've got that level of access, you're good.
I mean, for my, I just like, I can just like let them know to do it. I don't do this kind of stuff, but.
Yeah, that's
I'll give them your email and I explain like for fall, the sooner the better because we're very slow.
So,
this is Basil. I have a question. When you keep saying create an account, what does that mean? And when do you do that, or when don't you do that?
So, okay, let me go finish off on that question on unknowns and assignments a little bit, then I'll get to answer yours in a sec, okay? So if you remember so a little bit of history, in the old lab, we didn't have a back-end platform at all. And so in some ways that was easy, but it was also limiting because, you know, here we have an HCL unknown, but in the old lab, we had numbered unknowns, 1 through 15. And of course, no one's surprised by this, but those unknown results ended up on all of the cheating sites within a few weeks, right? And so to keep from cheating, right, we had to create this mechanism to create these unknowns, okay, that are not numbered, that get reported and then get saved into their lab book. So here is this unknown. So what they would do is come over here, put 1, 2, 0 and click submit, and then it ends up in their lab book. So we had to add that unknown. What Heather has done on those assignments, on those activities that you can integrate into your LMS, is she has built in some presets into those that use these unknowns, right? So you don't have to touch it. So this ability to go customize your assignments was for those customers who wanted that ability to customize what their unknowns were, the presets and all those things, and be able to do that we couldn't let them do in the past. So you don't have to use these assignments. These are just added features and functionality to customize the labs and customize your presets to whatever you want, okay? So just keep that in mind. You don't have to use the assignments. Those are just, like Heather said, customizable unknowns. So for example, if, again, see, what kind of user am I? So I'm an instructor. So for example, if I come over here to these assignments and I look at titrations right here, and I just look at the details here, notice here that on titrations, you can turn off the pH meter or turn off the conductivity meter or turn off the graph. So these are the kind of customizations you can do on some of these so that you can tailor this, because maybe you're trying to teach them how to do a colorimetric titration without looking at the graph or without looking at a conductivity meter or not being able to save the data to the lab book. And so these are just different ways that you can customize. All right. So,
and we're happy to like teach you how to do all that. That's really fun for us. But we're just trying to show you a good range of functionality here, and then we're happy to dive into those little teeny details of how you customize it for exactly what you want.
So basically, you asked the question about an account. So I have an account. So this is what a student would do, right? They would come over here, they would come and create an account. Because of privacy, we don't require first name, last name. You can ask the student to do that, but we as a company can't require that. They enter a username, that can be anything, an email address, but again, we can't require an identifiable thing. So just any username. We do require where they attend just so we can track. You put it in an email address so that we can verify accounts. You put it in, accept terms and conditions, and you register, okay? So that creates an account. Now, if I cancel here, and I'm gonna log in as some student here that I haven't registered, they won't have a code yet, okay? A student, so I don't know what the percentage is, half of the schools have their students purchase codes. So they would just go here and purchase a code, or the school can purchase codes. We would give you a list of codes, which you hand out to students, or bookstores can also purchase codes, which they can resell at in their bookstore, online or in person. Whatever it is, you're going to put in a code to give you access to the lab. So once you put in that code, you can have access to the lab. You know, you have profile here, and this one doesn't have any, unfortunately. So you can also kind of give student codes indirectly, and they can activate that way. So that's what a student would do. A student would create an account. They either buy the code, you give them a code, or they buy a code from the bookstore. They put that code in, and then they're activated. Then they can go use the labs. So we had lots of organic people. So I've shown some of the general chemistry labs as I was demonstrating different things. Let me kind of go to the synthesis lab and try that out. Here's, so again, pretty much the same. You can create, so there are no unknowns in synthesis, but you can create those presets where you can control certain things, which I'll show you in a minute. So here's our stockroom, no longer use here. One thing we've added, if you've worked with Kelsey, this was his idea. Knowing the equipment was hard. So you can mouse over the equipment and gives a little description of what it does, points to where it is, so students can learn where all of the equipment is in this lab. Here's our stock room again, where again, you pick a reaction. So I can pick this one and this one. And so works the same. Once you add those, kind of everything else will work the same. So I can come over here and add my acid. I can add my condenser and add my heat. I'm double clicking. You can click or drag or double click, and then boom, you can start it. And now you have live data. So this is one of the things you can control. In the old lab, this was always visible. You can create a preset where you can turn this off, so they can't see. So maybe they're going to give you some new reaction. You can't see what's in it, so they can't monitor that, and just cheat off it. So maybe all they would get is the TLC. So I can come over here, measure a TLC. Notice that this shows up over here on my, let me go bigger. You can click out of here. Let me go full screen again. And so this is temporary. This is just the ability to keep track of some of these things, but it does not go into the lab book until you save it. So if you click here and save it, then it will come over here into the lab book. You can zoom back into it, view it again if you want, or delete it. So this becomes a kind of like a reaction, like a staging area for things. So notice here that I'm seeing my products, and I can advance my time. Same thing here, and so I could stop anytime I want, pull over my SEP funnel. Now I have my aqueous reagents. Pick the one you want. Maybe I want to get rid of my acid, so I'm going to go double click. I add my sodium hydroxide. Here's my aqueous layer. Here's the organic layer. So I can pull this down. or I can pull it over here, it doesn't matter. I can extract one, I return it because I can't have them both there. Now I can do a distillation and separate it that way. So I can put over here, put on my nitrogen, and I can come here over here and start doing a distillation. I could still do TLC. Maybe I can come over here and just mouse over. It's nothing in the collection flask. So I'm seeing this over here, and I didn't make very much product. Come over here, separate it. So now I have it, and now you can do your spectra here. So here's your NMR spectra. You can zoom in. Peak numbers over here. Here's your integrations. Again, you can save this to the lab book. You can also flip this to a carbon-13 and do a carbon-13 spectra on it. And of course, FTIR, and save that. And you can do mass spec. So one of the other things that you can do, so you can see how all of this came over here. You can save that to your lab book. Those are the ones they want. So, you know, one of the things, again, that you can customize here besides the contents here is you can turn off certain spectra. So maybe you don't want them to look at mass spec. You can turn that off on one of these assignments, or turn off the FTIR, or turn off the NMR. We also have a spectral library. So we have a list of 750 spectra, and you can come over here and look at these as well. So you can turn these spectral libraries off, so they can't go look at that either. So, so this is what synthesis does. So you have a crystallization dish, you can do melting points, and we have all of these different reactions. And for those of you who aren't familiar with what this product does, you know, I can picking a reaction doesn't mean I have to do alkyl halide solvolysis. All it does is it picks what starting materials you're allowed to have. So you can pick 0, 1, or 2 of these starting materials. You can pick one of the solvents or no solvents, and then you can pick any combination of these reagents. So you can pick one of these reagents and the reaction conditions, and we're going to show you the kinetics, and we're going to show you the products that happen. So I'm not an organic chemist. I know esterification pretty well, but here's like the TNT reaction, right? So you can advance this. You can kind of see you're making your 2 different products, and you can keep on advancing your time. And so we have the kinetics of this. Color changes, we have explosions if they make mistakes. So you can do all of this, have ice, you know, for certain reactions. So the one thing that's new that we added is if the product, so this is, we had a constraint because obviously it would be it'd be a combinatorial problem that would be intractable. If a product that of one of the reactions is one of the starting materials, or you have 2 starting materials, is one of these starting materials, you can then take that product and do a next step and do a multi-step reaction. So that's new. The other thing that we changed is we did a much more accurate melting point for mixtures so that you can look at eutectics. So this is the synthesis lab. Somebody mentioned about, oops, I didn't want that. Somebody mentioned about organic qualitative analysis. We had all of this really good information on all of this spectra and all of this other data for all of our reactions. So we put together this qualitative analysis. So in this one, the whole idea is identifying compounds. So these are known. So these are the practice knowns that you can play with. And then we have unknowns. So every student would get an unknown here, and they don't know what it is. We don't show them the structure. They would get the same unknown every time they select it. So notice it's 179, 79.22. If I clear it, and I come back here and click it again, I'm going to get the same unknown, but every student gets a different one. So we have a database of about, 220 unknowns, and they can come over here and take this unknown, then they can take an aliquot, put it over here, we show them a picture, and then they can do functional group tests of all these different ones. So if I come over here and do Jones oxidation, then we show them a video. You can come and save this video to their lab book so they can keep a record of it. This is positive. And so you can dispose of this and come over here and do the next one. So you can do like permanganate test and go do this. So again, in the assignments, you can customize this so you can not allow them to see the boiling point or CH analysis. You can hide the spectra. You can also hide and turn off the different spectra here so that you force them to go through the functional group test. So, and again, all unknowns do the same way. I would put in, I know it's not acetone, but you submit it and then they get their answer, you come to their lab book and then you see their answer down here. And then you go through and grade it. So this gives you what the 2 organic products will do. And so I think we kind of shown what the whole thing can do. And let's say that they want to go to section 2, right? So what you would do is, this is the actual code. I'm just copying it with this. I just copied it to the buffer. I could also copy it this way, but that's the code, okay? And then if I was a student, so good question, I'm glad you brought it up. And I came in as, let me go see if I can find a student that I haven't joined anywhere. What they would do is they'd come to groups and they would click, oh, I'm already on one. I would come over here and click join a group. I'm gonna change this to 1 because I'm gonna have them join the other one. And I would click done and then they join the group. So now they're in the group and they don't need to worry about it. That's it.
And that lets you then see their lab book from within there and they'll be in the right location in your lab book.
Yep. Yeah, so that puts them into the group. And then for you, then, right? So if I come back in and sign in as my instructor, for you, like if you come over here to lab book, I have a filter. I can say, I want to look at all groups, or I want to look at just section 1, or I want to look at just section 2. Then you have this filter to go filter out what groups you're looking at. Same thing over here on assignments. You know, you can look at all your groups or just look at the assignments you have for group 1 or just the assignments for group 2 and so on. Obviously, I'm teaching 2 sections of Chem 105 this fall. I'm lumping them all into one big group because they're all getting the same grades from the same group. But you can
set it up however you want.
However you want, yep. I do want to show you something cool for those of you who teach PChem. Or kinetics, or this is a simple kinetics reaction is, so this is quantum, I didn't show it, but let's take a source like a super light bulb and I'm gonna pick liquids. We've added crystal violet as an option here. So we have different concentrations of the crystal, of different concentrations of OH with the constant concentration of the crystal violet. And so you can add that. And I can come over here and detect that with my spectrometer. So I can come build this. Of course, we have to write some content for this. We just haven't written it yet. But I'm going to turn on my light bulb and turn on my spectrometer. And there is the crystal violet spectrum. I can go to the visible spectrum if I want. And I can come over here and zoom in and just zoom in on this. And so if you notice, this peak is changing height because it's reacting. And so the way the student would collect their data is you can come over here and collect screens, save the screen to the lab book at certain time intervals. And then they could come here to the lab book and analyze this. and be able to pull the height out and be able to go do some kinetics experiments. So we're pretty happy about that. And if you've got more questions that you want to discuss in depth, join again and we'll help you out.